Okay, I'm going to tell you today about the work that we are doing in single-cell transcriptomic data analysis and development of gene signatures to identify specific cell types. That is a typical problem on single-cell analysis. And this is focused on the analysis of hematological and immune cells, because we work with cancer cells and tumor, and we try to investigate the complexity of the tumor and how the different cell types, immune and hematological, are involved in the tumor environment. But we started to do the analysis on the specific on the hematological and immune cells, that, as you know, is a complex system that we have in all our health system, our blood, and the development of these cell types, like is quite complicated, where there are many cell types and subtypes, and it is important to identify the different subtypes, for example, different subtypes of B cells, or T cells, or dendritic cells, and how we do that with the single cell data. No? So what we want to do when we apply a technique like single cell data analysis or single cell technique, and the analysis of the data derived from this technique, is to resemble what is known in nature, what is how is the composition of our real blood or peripheral blood. And for example, here you see that when we are talking about whole blood, is not the same as other parts of the blood. That here, for example, the number of neutrophils and eosinophils is the largest percentage of around 50 in terms of cellularity, nuclear proper cells, no, even depleting of erythrocytes. 57%. So most of the studies done on hematological immune cells are done in peripheral blood mononuclear cells. That is another subset of this part of our blood, of our bone marrow. And you can see there that again, this part of the blood tissue, if you want, the PBMCs, are depleted of granulocytes, especially all the basophils and neutrophils. So sometimes depend on the nature of the sample that you are analyzing to resemble what is happening in hematopoietic cell production is not easy, and to distinguish all these different cell types and subtypes. For example, we were talking the other day about the different subtypes of dendritic cells coming from the lymphoid lineage or the myeloid lineage. And also we are collaborating in a COS project, because many times, many studies, when we study hematological or immune cells, the myeloid lineage is a little bit not considered, because the granulocytes are not included in many single cell data sets, or the monocytes and macrophages are many times confused with dendritic and things like that. For example, this is a science paper derived for a deep analysis on just on dendritic cells, and here this paper of several years ago is characterizing 6 different types of dendritic cells. So you realize that, and it's a very small one, around 2% of the mononuclear cells. So I'm going to present the study of 2 of my students, Natalia and Enrique. The first part of Natalia is that we are working on the development of methods and algorithms for do analysis of bulk expression data, to analyze cell mixtures and deconvolute and calculate the concentration of the different cell subtypes. That this is a problem because we have, well, it is a challenge because we have many bulk RNA-seq data from many samples in tumors in cancer that we are analyzing. So we need to be able to deconvolute, to extract the information about the different cell subtypes on this kind of data, that is bulk data. And to do that, we need to do 2 things. One is to have an identification, a sort of signature, a gene signature matrix for identifying the different cell types or cell subtypes, and also, in this case, the bulk data. No? Many times the gene signature comes from single cell data, and this is overimposed in bulk expression data. And what we have done here with Natalia is to try to apply several methods of the convolution to see how they perform in the identification of the different cell subtypes. And we have done this with PBMC data with these 4 data sets, and that we have different concentrated numbers of cells, and how the different methods are able to identify different cells. And quickly in this part, what I'm going to say that the first challenge when we approach that is that the granularity of the different studies is very different. And this is a big challenge, because as you can see, we are analyzing here 4 data sets, and the cell granularity, for example, for lymphocytes T cells, is, if we are considering the first data set, has got 17 different cell types, and of T cells, there are 7 different T cells considered, but other data sets are not having the same granularity. Granularity means how many different subtypes or cell subtypes you are distinguishing, 17 or 11 or 5 or 4. CyberSort, the data matrix of CyberSort base that is in standard not to identify cell subtypes, immune and hematological cells, is 22 different cell types. So where are you in the granularity when you want to do a single cell analysis? No, we compare different methods. This is more about algorithmics, but what we can say that, for example, CyberSort, when this is a data set where we have the reference, because it's a produced data set where they have put a certain amount of tumor cells, a certain amount of B cells, T cells, NK cells, and so on. And we know what is the real mixture there, how the methods perform. For example, in this kind of CyberSort, you can see that the real is the real or known expected column. CyberSort, despite the 2 first columns, sample 1 and sample 2 correspond only to tumor cells. And you can say that CyberSort is estimating the presence of immune cells, despite the fact that in that sample there is not any immune cells. So, and this is the same here with another method, the comparison with Linset, another method that is a non-supervised method. CyberSort, again, here you can see sample 1 and sample 2, they have only 2 cell line. Those are colon cancer cells that are produced in this sample to do the comparison of the algorithms. And then sample 2 and sample 1, they are only tumor cells without any other of these immune cells. But again, CyberSort still is estimating the presence of certain immune cells. Why? Because it's designed to estimate immune cells. So what we managed to do after all this work basically is to create a signature, is an optimized signature, little signature, through this method, Linset, that is non-supervised. It doesn't need a preliminary signature. Linset produce in the process of running the algorithm, produce the signature for the cells that it is identifying. For this identification of neutrophils, monocytes, B cells, T cells, and NK cells, the Linset produce a gene signature of 170 genes that performs better, or mostly similar, but in this case of better, as you can see, than CyberSort. So this is a first question that we have to address when we go into, and this is the paper what we publish about, how we apply different methods of the convolution for identify different cell types, especially in blood, immune, and cancer cells. But once we run these methods, yes, this method is a single cell. We want into a single cell development of a single cell workflow to analyze single cells, and this is done by Enrique. Then this is a complete workflow that we have developed for the analysis and identify of different cell types and subtypes. We did the same, the development of this algorithm, of this workflow, sorry, is a combination of algorithms, it's not our workflow. What we apply this again to PBMCs mainly and data, and to the three main data sets. In one of them, data set one, we have all the known, preliminary known concentration of the different cell types done by flow cytometry for this data set. So we have a reference. So again, what is the first promise when we go into this analysis is at what level of granularity we want to do the analysis, because, and this is a table that we prepared because we can do the analysis of these three single cell data sets at three levels, considering the number of levels, as you said, of types, let's say 7 cell types, or 16 cell types, or 27 cell types. So it depends on the subtypes that you want to identify. And the complexity, of course, is bigger when you go into deeper granularity. So in this way, we analyze all this data. This is heavy. Okay. Go. No. Es que no pasa porque pesa mucho. Ya está. Thank you. Well, those are the different single cell analysis that we did for this data set, and you can see the granularity for the first data set. We are investigating 27 cell types, or 19 cell types, or 12 cell types. And what is the three panels, the three lists, it depends what are the gene signatures or the gene markers that we are using. So in a way, we are evaluating this, the power of the different signatures. One signature that we used by default was to take the 369 CD markers that are well known to characterize most of the cells, but we also took membrane proteins and all the genes expressed in the system, and so, and let's see, let's go. Sorry, this is too heavy, too many points. Well, what we did is to apply these different signatures, and then we ran a machine learning method, the random forest, to identify which were the signatures that better identify the different cell types. And this is what I wanted to show you, if it is possible. Sorry about this. Too heavy. Oh, this is a silhouette algorithm to identify the groups, the cell groups. And then at the end, we managed to identify several signatures for each cell type. And what we do here is we run the random forest to identify how is the performance identification of the different cell types at different levels of granularity, 7, 17, 16 or 27, and then how is the performance. And we identify the signatures that perform the best with this random forest approach, and then these cells are identified. So basically, later on we do on our map, sorry about this, we do a trajectory analysis to see if the well, this is not performing, it doesn't matter, but here you can see our map here. In the map is the 27 different cell types, and you can see that we can follow the myeloid lineage in the evolution from stem cells, hematological stem cells, to monocytes, classical monocytes, or non-classical monocytes, and so on, or the lymphoid lineage, or the lymphoid lineage to B cells, or the lymphoid lineage to T cells, and this is very well matched according to the signatures that we managed to get with this trajectory. So this is the work that I wanted to present to you, that we are working to this analysis of single-cell transcriptomic data in the trying to identify good gene signatures in the identification of the different cells and cell subtypes. And all the people that work in different projects in my group and collaborations, and here the main work that I presented is by Natalia and Enrique. And thank you.
Yeah. This is very impressive how you can compact all of this. All right.
One question?
Only one. Oh, it was too much information. Yeah. One, only one.
Thank you for your presentation.
Yes.
Thank you for your presentation. And so these results that we observed in the presentation were observed in a liquid sample. So I was wondering if in tissue or solid sample, the results would be similar according to the error that CyberSource is showing. So the nature of the sample can affect the estimation made by CyberSource.
Well, this is a good question. Yes, it's true. We work a lot for this work with hematologists and in cancer, hematological cancer. So this liquid is not solid tumors. It's true that in solid tumor, the identification of the different subtypes can be a problem, but the problem is not it by itself. The main problem is in the processing of the sample, because you have to detach the cells and then put into the single cell machine, the 10X or whatever, and then this is a problem that is due to the separation of the cells. How do you do trypsinization or some sort of treatment to detach the cells? In this process, you can damage, of course, the number of cells of a given type, or so and so on. But of course, the analysis of single cell tumor, solid tumors is different. But for our approach here is identifying the different cell subtypes. So for this reason, these liquid samples are better to have as well, because we can have reference data where through flow cytometry we know what is the real concentration of a single cell. So this is not so easy to do with tumor, but we are going into this problem.